Hi guys, we're going to talk about cell transport today. So I'm sure many people wished that things worked the way they do in this cartoon, where basically you could use transport proteins to help get you out of any enclosed space. Unfortunately, not quite the case. But let's take a look at how cells transport things in and out. So a few definitions and things to go through first. If a substance can pass through a barrier, we say that barrier is permeable to the substance. Um, and so, for example, if we're talking about a cell membrane is permeable to O2 molecules. If a substance cannot pass through a membrane, then we say the membrane is impermeable to it, meaning it does not allow that substance to pass through. So the membrane is impermeable to glucose, it's impermeable to ions. And then we say that the way we describe most membranes is that they are semi-permeable and they allow some substances to pass through, but not others. Another way to say this is that these membranes are selectively permeable. So, selectively permeable or semi-permeable, that is our descriptor for the cell membrane because even when it's just a phospholipid bilayer, it is going to allow certain substances to cross and certain substances will not be able to cross. So we can say the membrane is impermeable to these molecules, but the membrane is permeable to these other molecules. Also, we'll be using the term solute and solvent quite a bit. Solutes are any molecules that are dissolved in another liquid. That liquid that stuff is dissolved in is called a solvent. Here in biology, we are only going to talk about one type of solvent, and that is water. And anything that is dissolved in water, we call that type of solution an aqueous solution. An aqueous solution is one that has its solvent as water. So your entire body is made up of various aqueous solutions, in general, separated by membranes and other barriers. Um, and then the important thing is that because we have these barriers, we actually can form what are called concentration gradients. So some of these exist naturally, some of these your cells make. What a concentration gradient is it's a difference in concentration of any substance across either a distance or a boundary. So in this picture that we see here, we have a high concentration of these red molecules on the top and we have a low concentration of these red molecules on the bottom. We have a difference in concentration across a barrier. Therefore, we have a concentration gradient. Now, naturally, with no outside energy input, molecules will move down their concentration gradient, by which we mean they will move from high to low concentration. And the cell doesn't have to use any energy to make this happen. This happens on its own. That's diffusion, if you remember from freshman biology. Another thing is one common theme we'll see in the future is that cells often will create a concentration gradient. So, for example, here we see this protein, channel protein, is actually going to use energy represented by this lightning bolt to actively pump these red molecules from a low concentration to a high concentration. So they're using energy to do that, and since those molecules, if they are allowed to move on their own, will naturally move down their concentration gradient, you can use their diffusion to then do work. And that's how a lot of processes in the cell, especially cell respiration and photosynthesis, happen. They rely on the cell building these concentration gradients and then using those concentration gradients, the energy that's stored in them, to do work. So concentration gradients can store energy. So right now, you have potential energy in this system because there's a difference in concentration. If I were to open some sort of gap, um, pretend like that's a gap in the membrane, then these molecules would be able to diffuse down their concentration gradient. And then I could use their movement across the concentration gradient to do other work. So here we have just a stereotypical phospholipid bilayer. Notice there are no proteins or anything else stuck in it. And I tried to get this kind of to scale, but it's not quite. In reality, glucose is much smaller than a phospholipid, and oxygen and hydrogen ions are even smaller, but I wanted to make it so that you could actually see things. So we've got purple oxygen atoms, yellow hydrogen atoms. When you have this lone hydrogen atom, like you see on the bottom, that is a hydrogen ion. And then I've also labeled the parts of the membrane as being either hydrophobic or hydrophilic, because remember, the heads of phospholipids are hydrophilic and are polar and are therefore attracted to water. And the interior or the tails are hydrophobic, they are nonpolar and are therefore repelled by water. So, what kinds of materials can pass through? Well, we traditionally talk about water passing through, but if you remember, things have to be small and they have to be uncharged in order to pass through a membrane. Basically, if it's large and in charge, it cannot cross. And water does have charges, it is polar. And while some water can go through a membrane, the rate at which it can pass through a membrane is super slow. So, the like phospholipid bilayer by itself is only slightly permeable to water. And we'll talk therefore about how water gets across cell membranes more a little later. A small uncharged molecule like oxygen can actually go easily through. So that's very good for your cells because you need oxygen to enter your cells. You also need carbon dioxide to leave your cells. And that can happen without your cells doing anything because they will simply diffuse across the membrane. Larger molecules like this glucose will not be able to cross the membrane. They're too big to fit in between the spaces. And then super or highly concentrated charges like ions will also not be able to cross the membrane because they are repelled by that hydrophobic layer. Or you can say they're attracted to the hydrophilic layer. So that's what can cross the membrane. If it's large, like that glucose molecule, or charged, like that hydrogen ion, it will not be able to cross. However, so that's what it's saying there. However, if you introduce something like this, a channel protein, that channel protein can change the permeability of the membrane. And that's actually what makes all the difference in a membrane. So you still, this channel proteins are usually fairly specific. They will let certain molecules in. So this guy is a glucose transporter. It will specifically allow glucose to enter and transport it across the membrane. So we see glucose, it enters, and then it ends up coming out on the other side. Um, so the channel proteins, the various proteins that you have embedded in the membrane are going to be very important to determining its characteristics. What can cross it, what cannot cross it, what it's permeable to, what it's impermeable to. So you'll have distinct regions of membrane in the cell. Your cell membrane may have different proteins in it than, say, your outer mitochondrial membrane or your inner mitochondrial membrane or your Golgi membrane. And it's the characteristics of those proteins that are embedded in the membrane that are going to give the membrane its specific properties. So let's take a look at this just in summary. You have aqueous solutions both inside the cell and outside the cell. And remember, we have that nice hydrophobic core inside the middle of the phospholipid bilayer. So charged molecules and ions cannot cross the phospholipid barrier on their own. You do have channel proteins that we'll talk about in a little bit that are there specifically to allow water to get in and out of a cell. Small and non-charged molecules can cross the phospholipid bilayer. But if it is a large macromolecule like this protein that's here, it will not be able to cross. So let's talk about different types of transport. This is passive transport. So what this dude is supposed to look like is like he's just kind of lying on his back with his legs, just kind of bent and spread out like he's just chilling. Passive is like that. You don't really use energy for it. It just kind of happens. So passive transport does not require the cell to use energy. Basically, it does not need to use ATP. This happens spontaneously without the cell doing any work. So anytime you have passive transport happening, the cell does not have to expend energy to make this happen. And it always involves the net movement of molecules from a high concentration to a low concentration. That is one of the defining characteristics of passive transport. Without any other outside changes to the system, these molecules will move until they reach equilibrium. Equilibrium is when you have equal concentrations, either throughout an area or across a barrier. And while molecules are still constantly moving, if you remember from chemistry, as long as you're not at absolute zero, you are still in motion as a molecule. While these molecules are constantly moving, there's no net movement, meaning for every molecule that moves, say, from this side of the membrane to this side of the membrane, you will have an equal amount move across the membrane. So it's like the individual molecules may move, but the total concentrations stay the same. Very similar to how in times of COVID, if you're at a restaurant or if you're at um, a gym taking your senior pictures, they will let certain amounts of you inside at a time. So if only 20 people are allowed inside at a time, the identity of the individual people who are inside that space may change. May not may change, but the total amount of people inside will not change. So nobody's allowed to enter until one person leaves. So that's kind of how equilibrium works. So there are multiple types of passive transport. There is simple diffusion, and this happens only when the membrane is permeable to this molecule. There is facilitated diffusion, and these two middle examples are two types of facilitated diffusion, one of which can be just a channel that allows certain ions or molecules to cross through. Another can be an actual carrier protein or a transporter. And what has to happen is this thing has to change shape for the molecule or whatever it is to be transported across the membrane. That's what happens with glucose. Now, this shape change does not require the cell to use any energy. When a glucose molecule binds to this transporter on the outside of the cell, that binding makes the protein change shape spontaneously to this shape. And that shape happens to have low binding affinity for glucose and also opens to the inside of the cell. So it transports things, but then the cell doesn't have to use any energy. Once this guy has released its glucose, that spontaneously makes it change its shape back to the original conformation that's open to the outside. And then I said we would talk about the specific case of transporting water. Very little water will be able to pass through the phospholipid bilayer. That's why that's kind of a dotted line. However, you have these specialized proteins called aquaporins, whose entire job is to allow water to cross the membrane. It does not dictate what direction water can cross, it just allows water to cross. So, once again, the three different types of passive transport. Simple diffusion happens with uncharged or small molecules. If it's small and it's uncharged, it can cross through simple diffusion. If it is large or charged, you will need facilitated diffusion, or you may even need a form of active transport. Things like glucose, water, ions require some sort of transmembrane protein that goes across the membrane. And then osmosis is the very special case of diffusion of water, and it's aided by aquaporins, which I will mention again later. So let's look at simple diffusion. These are small uncharged particles. They can move freely across the membrane. And unless you have like some external source of these molecules coming in, or unless the cell spends energy in order to make this not happen, these molecules will reach equilibrium. So we start out with this panel right here. You have a bunch of these molecules that are located on the um, outside of a cell, and very few, or if any, inside the cell. As time passes, more of them are crossing through, and they are crossing until there's approximately equal concentrations on each side. Now, things are still moving, so this guy may cross the membrane that way, but then this guy may cross the membrane going in the other direction. And while individual molecules are moving, there's no total or net change in concentration. Facilitated diffusion, like I was saying before, can either happen with just a channel which has a hole in the center and will allow certain things to cross. It doesn't necessarily allow any any old molecule or any ion to cross. Some of them, especially if they're letting ions cross, will have specific charges or specific shapes to specify which molecules can cross. Carrier proteins, they change shape to pass things across the membrane, and their change in shape does not use energy. It happens when they bind to whatever they're passing across. So here we see the glucose transporter, which is really important because while you want glucose to be able to come into the cell, it's too big to pass through between the phospholipids. So you have these glucose transporters in the cell membrane. So here we see a glucose that's outside of the cell, and the glucose transporter is just in its normal unbonded conformation or shape where it's open to the outside of the cell. Next, a glucose molecule binds to it from the outside. That binding makes the shape of the transporter change. It changes its conformation, its shape. The change will do two things. One, it will make the transporter open to the inside of the cell, and two, it will make it less attractive to glucose. So glucose will be only weakly bound, which means um, once the glucose is kind of facing towards the inside of the cell, it'll be released by this transporter, and then that release causes the transporter to change back to its original shape. And then it's ready to bind another glucose, change shape, and allow glucose to enter the cell all over again. So notice there is no energy being used. All of this happens by the binding or the lack of binding of glucose. So water is very important to allow to cross cell membrane boundaries fairly freely. And what lets them do that is aquaporin. What you see here is a 3D model of an aquaporin that is stuck in a cell membrane. Those ribbons are basically alpha helices, and that's kind of showing you the general shape of the protein. It does have a hollow core in the center that's got charges that will only allow polar water, so specific shape and a specific charge to cross through. And it will allow lots and lots of water to pass through. And you can also control how permeable a cell membrane is to water by increasing or decreasing the number of aquaporins you have in your membrane. You need to let more water cross, put a bunch more aquaporins in your membrane. That is not what I meant to do. My bad. Oopsie. All right, so osmosis is the diffusion of water, and this happens mostly, or this is most noticeable, when you have solutes that cannot cross whatever barrier is separating two things. But the solvent, water, can. So if we were able to set this up, and I've never been able to get this to work, but if you were able to very accurately and firmly separate a container into two different sides by a membrane that's only permeable to water, and you put two different solutions in, what you should see is that water will move towards the more concentrated side, and the water level should change. You should have noticed that in the pivot that you guys were doing, the pivot lab. You should have noticed some water levels changing. And we're going to talk much more about the movement of water in another lesson because it gets complicated. So now active transport, like this dude who's now actively up and running, it requires energy. So this is where a cell has to use ATP. And in general, it's moving things from a region of low concentration to a region of high concentration. So we can say that it moves things against the concentration gradient. And for this, it's going to be using transport proteins, transmembrane proteins, because it's moving things against the concentration gradient, which takes energy and effort. It's not going to happen just straight through the membrane. So the most common form is a pump. A pump is going to use ATP to move molecules. So here, what we see pictured is a proton pump. This proton pump is using energy to actively pump hydrogen outside of the cell. Keep in mind, we have a low concentration inside the cell, and a high concentration outside the cell. So we are moving from a low concentration to a high concentration. We are also using ATP, and both of those things let us know that this is active transport. And you may ask yourself, well, why would you be wanting to move around protons? That's going to become really important for photosynthesis and cell respiration. You also have the sodium potassium pump, which you may have looked at briefly in freshman bio. Let's look at it in more detail now. So the sodium potassium pump is its function is to build a concentration gradient. What it is doing is it is making sure that you have a high concentration of sodium on the outside and a low concentration of sodium on the inside. So what it's doing is it's actively pumping out sodiums. So these guys are sodiums, the hexagons. These guys are potassiums, our ovals. And so sodium is basically being pumped out of the cell. So you see these sodiums here, they are binding to the sodium potassium pump. Then you use some ATP, which is coming in right here. That ATP is used to change the shape of this protein, which makes it less attractive to sodium, so those sodiums get spit out. And it also makes this protein open to the outside of the cell. Once it's open to the outside of the cell, the change in shape makes it much more attractive to potassium, and the potassium can come in and bind. That potassium binding triggers a shape change, which will do a couple of things. One, it's going to cause that phosphate that was attached to it from the ATP to release. When that phosphate is released, that's going to make this sodium potassium pump change shape. When it changes shape, it is no longer going to effectively bind to potassium, so those potassium ions get spit out. And they're going to get spit out to the inside of the cell because that shape change makes it face the inside of the cell again. So you're building up a concentration gradient. You want high sodium on the outside, low sodium on the inside. You want high potassium on the inside, and you want low potassium on the outside. And my bad, that's supposed to be K. I don't know why I'm writing phosphorus. Okay. So what is the point of doing this? Because back when I would learn about the sodium potassium pump before I, went into neuroscience, I was like, okay, what's the point of this? Well, the point of this is that when you have these different concentrations of these ions across a membrane, you can then open some channels, let these ions cross, and ions carry charges. So you can have moving charges. Moving charges create an electrical signal. We say that this charge difference that you have across a membrane is called a membrane potential, kind of like potential energy. It's voltage potential. And so the sodium potassium pump builds up a concentration gradient of sodiums and potassiums, and the neurons in your body, as well as your muscle cells, will use this difference in concentrations to send an electrical signal along a neuron and carry information. And that is actually in the form of what's called an action potential. um, we no longer cover that in AP Bio, and I don't really have time to get much into it right now, although I'll probably make a video later about it, just because, it's neuroscience, it's my jam. So you have in this diagram a negative net charge on the inside, a positive net charge on the outside, and this charge difference means that the membrane potential of this cell is around like negative 70 millivolts. You don't need to know that number. You just need to know it's negative. So when you have channels open, sodium, which has a high concentration on the outside, will actually rush into the cell, and that will make the cell more positive, which will change its membrane potential. That change in charge, or that movement of charge, is electricity. That's all electricity is, except in electricity, your moving charges are electrons. So, like I said, I'll try and do another video later to go over that. Now, the other thing that uses a lot of energy is bulk transport. This is most definitely not going to happen spontaneously. You've got a number of proteins that are going to make this happen. So, bringing stuff into a cell through bulk transport is called endocytosis. Endocytosis is going to be basically, um, in this case, in the diagram, you've got a pseudopod, which means fake arm or fake foot, which is basically these extensions of the cell that kind of reach out and wrap around something, and then they will enclose it in a vesicle. So this will pinch off and form this vesicle like you see here. Here, you also see that process happening. You see kind of, but instead of arms reaching out, you see this dip in the membrane, almost like someone's pushing into it. And then as it kind of stretches, this part of the membrane gets joined and forms this vesicle that's coming inside. Very similar process happening here, except it is basically triggered by the binding of a molecule to this receptor that triggers it to form a vesicle, and that's another form of endocytosis. By the way, that process of kind of like making that dip or that inner pouch or depression in the membrane is not only called infolding, it's also called invagination. So if you see that term, that is referring to that membrane kind of basically bubbling off to form a vesicle. And so that is bringing stuff into the cell. And depending on what you're bringing in, that's going to determine the type of endocytosis it is. I don't anticipate you necessarily having to know these, but I do want to make sure that you know that phagocytosis is when you're taking in solid particles. This is how a lot of single celled creatures eat. They will endocytose something, bring it in a food vesicle, and use a lysosome to help digest that. Penocytosis is when you are just taking in some of the extracellular fluid. There might be some molecules in the extracellular environment that you want to take in. And then receptor mediated endocytosis is triggered by the binding of a certain molecule to a receptor on the surface, which then causes endocytosis of that area. And it's not really emphasized. I did not put it in here because I did not want to confuse people, but this fuzzy stuff is actually what's really found on these vesicles. It's called clathrin. And so these are like clathrin coated pits, basically. And clathrin has a job in helping to form vesicles. Exocytosis is the opposite process. So you start with the vesicle inside the cell. That vesicle will come up to the membrane, and its membrane will join with the cell membrane. Once it joins with the cell membrane, it becomes continuous with the cell membrane, and whatever was inside that vesicle gets released. So, any cell that really does any secretion is when you're like releasing molecules out of the cell. Those are going to be usually doing this by exocytosis. So they'll make a bunch of whatever protein or molecule they want to release. They'll pop it into a vesicle. That vesicle will go up to the membrane, basically melt into it. That's kind of how I visualize it. And then that will release the contents of that vesicle outside of the cell. And that's a very common thing that happens. That happens in neurons with neurotransmitters being released. That happens in cells that are producing a lot of proteins for export. So, say a cell that's producing a protein hormone. And then the membrane that made up the vesicle becomes part of the cell membrane. So that's also another thing that can be transported. You can have the molecules, the proteins that you want to have embedded in the cell membrane in the walls of these vesicles because that vesicle is going to form part of the cell membrane. So you'll have membrane added to a cell's plasma membrane by exocytosis, and then some of it will be taken in by endocytosis. Because if you just kept adding membrane to a cell, you would just keep making it bigger and bigger and bigger. So that is it for this lesson. Next up is going to be a lesson on tonicity and osmoregulation. Just as fun as it sounds.